Okay, can you hear me? Anybody can hear me?
Yes, we can hear.
Yes, ma'am.
Finally. So then we can try to start. Third time pays for all. Just a minute, I need to close the door, otherwise we are going to see. Okay, so then we are going to start with our first lecture of the course, which should be called Theory and Practice in Biological Experiments.
You guys calm down. You guys calm down. At least if everybody will be against us. Don't be we when we see if he do anything. Since they admit say don't do mistake like this. Okay, if they talk say this one. Don't think we can't take different from what is heartless government. And what they admit say when a mistake, put it to delete it. The least we can do.
I'm going to mute all of us. If you could, please, because when you speak, I'm not really able to hear what you're talking about, and I'm not really able to hear what I am talking about. So if you could please then write to the chat, this message board, or please then ask questions in written form, because otherwise it will be very complicated to communicate, since we are kind of 9 of us. Is it okay? Hope it is okay.
Yeah. Yes, it is okay.
It's okay, ma'am.
Okay, then I'm just starting. We'll try to hold and keep all our questions in the very end, but still, if you have something important, please just say beep and we can discuss the question. So, in our laboratory of molecular biology, neuroscience, and bioscreening in MIPT, we are going to talk about theory and practice in biological experiments today, hopefully. So to start with, we are going to discuss what is an experiment. It works. So what goes for an experiment and what you're going to have from the experiment. So experiment is any action that goes through the test, and when you test any hypothesis, this is an experiment. So for instance, when you have a child and it tries to investigate whether the round shape fits into the square hole in the toy, this is the first way of doing experiment. So what you gain from an experiment, you gain an answer. But to gain an answer, first you need a question. So to conduct an experiment, you need to have primarily a theory. If you do not have a theory, you cannot ask a question, and then you cannot really have an answer. To start with, you have many types of experiments, and we can distinguish three types of those. So virtual is when you just imagine a model and you think whether your theory or your question have an answer in common. Is it possible to get this answer? And then you have to imagine all the details of your experiment, because if you do not have the plan how to conduct it, then it will be spoiled 90% of probability. So then you have a pilot experiment when you test in the reality, on practice, whether your method and what you have imagined is working or not. So pilot experiment is something with low statistics or no statistics at all, because then you just test your methodology. And then you have your final row of experiments when you have your statistics and answers, and you have your yes or no, and you have to repeat it many times to be sure that the question is really answered. So basically, good experiments lead to the result, and to conduct this experiment, you have first to imagine a theory, because only then you are able to ask proper questions. To ask proper questions, you have to know more than half of an answer, because otherwise your questions are not really good. Sorry, I have to add guys to the lecture. So we're 11 now. So this is the first part of your thinking. You have to be really good in thinking to start the experiment. And normally it should take you kind of half of your experimental time to think about your experiment. Then, suppose we have a theory, then the purpose of the experiment is to test this theory, and then what is the problem and what is the aim of the experiment. So to formulate the aim of the experiment, you have to divide the theory into small pieces, and then one theory piece should be proven by one element and one experiment. So if the example is the Earth is round and it goes around the sun, then you have already 3 pieces here, because first piece is that the Earth is round, and then the second piece is that it goes. And then around the sun is the third one. So you have to conduct the experiment for every piece of those to prove those separately. And if you're good enough, then you have to run the final experiment to prove that all 3 are taking place at the same time. So the task you're running and the aims of your experiments, they determine the results of your experiment. So that if the tasks are bad, then the results are bad. If the aims are not good, then the results of your experiments are not really good and they are not publishable, and you are not good with those as well. So the results of the experiments, they may be qualitative or quantitative. And this is the difference, because for quantitative, you need the good statistics. For qualitative, you don't need good statistics. You just need a proof that it goes or it doesn't. Suppose we have a theory and we have a good problem, and what can be the result of the experiment? Then, well-defined tasks lead to the results, but what results are good? The results are good when they are achieved during the run of the experiments, and they are reproducible, and they are valuable for a theory because they have to answer your question, and they are suitable for further analysis. So there should be something more than that result running, and there should be an application, because otherwise you don't have funding in most cases. So if you do not have all these 5 points, then the task and the aim is probably not really good, and you have to go step back and analyze whether your plan of an experiment and whether your virtual experiment has any problems and whether it could be improved. Because if the results are poor, then you lose a lot of time to perform an experiment and to measure the things and then to calculate them, and then you have no results. So the way you plan your experiment depends upon the tasks you solve, and it also depends upon the results you think you're going to have. So within the framework of the problem, what should be the plan of experiments and what will lead to the most reliable and reproducible result? Then when you think about it, you have to take into account all the factors and parameters in your experiments that affect the experimental object, and think about random disturbances and random events that can influence your experiment. For instance, when we started the lecture, the talk, I went away to close the door, because this is how I reduce the amount of the spontaneous interruptions. And therefore, this makes my experiment a little bit better. So all the experiment parameters, they should be connected to the controls you are going to run. Because if you just run the experiment without control, you'll have problems with proving whether your experiment really demonstrated an answer, whether there was an answer, whether the answer was good. So experimental parameters, they can be divided into random, that you cannot really control, and controlled, which you can fix and vary. Usually you focus on the fixed and variable parameter because this is the body of your experiment. You have something and then you have like something to influence and then you have the result. So in most cases. Or you have something and then you have treatment and then you want to see the result. This is also something you control. What you do not really control is when you have a system with something inside that behave differently from your expectations, or when it's technically independent from your brilliant mind. So, for instance, if we have an electricity shutdown, this is random, and this is independent from your will. So you have to think about it when you plan your experiment. Or something like your colleague came into the room and took some of your, I don't know, valuable buffers and did something. And this is random. It cannot be controlled unless you close the door and turn the key. So when you start to plan your experiment, you have to think twice at least. What are the controlled parameter and what are the random parameter and what parameter you can really control. So to be sure that you are good with controlling and to be sure that there were no random influences, you make controls. So controls can be divided into repeats and controls that really prove something apart from your main influence on the experiment. So repeats can filter out random disturbances such as electricity shutdown or colleagues running into or something unexpected like earthquake. Controls allow you to take into account third-party factors and something that you should have thought of, but you cannot really control it, or you're not really sure whether you can control it. So from this point of view, when you have your experiment and you have your parameter, they can be controlled manually, like flashes in the sun, they cannot really be controlled. But gas composition, humidity, failure during experiment, all this you can filter. Also, when you have control parameter, this is, for instance, light wavelengths, intensity, concentrations or density of your cells or whatever. So when you divide this parameter, you have to be clear in your mind what is controlled and what is not. Here we can talk about whether the experiment you plan is going to answer the question you really asked. So if you have a theory and then you perform some kind of experiment, for instance, under the influence of green light, bacteria, any kind of, they change their color and you want to prove it. So to check if the bacteria change the color when exposed to the green light, what you do, you make an experiment. You irradiate bacteria with green light and then check in few hours and then you have your answer. So here you have two parts of the answer. First is whether bacteria really change their light color. And then the second answer is when they really change their color. So this experiment gives you single result and you have no doubts about this result. If the experiment is not really good, we just skip a little bit further. So, and you make your control and you use the ultraviolet light to be control like for the green light. So when you have your red bacteria and you expect them to be blue and you apply green light, okay, they're blue. But when you have red bacteria and then you apply ultraviolet light and then they're still red. What I want to hear from you is whether this experiment is good. And if it's not, then why it's not good? Because it's not for the experiments, in this case, it's for the controls. If we go back, then in common, your experiment plan of thinking, the pipeline should be the theory, and then the aim, and then the task, and then the results from your imaginary experiment. So you have a kind of prediction, and then from this prediction you have to gain the parameter you're interested in, and based on this parameter and your prediction, only then you can go to the layout. Because usually you start with layout, and this is the problem of starting your first independent work, because you have the hints from everywhere and you are not really able to stop and think whether your layout is really good and whether you do really understand why your layout goes like that. So for this layout composition, you need to think about basic pipeline and then controls and then repeats. So the basic pipeline is how you are going to perform the experiment. The controls is how you're going to prove that parameter you've changed and the parameter you didn't change, influenced or not influenced on the results you've gained. And then repeats is to gain the statistics and to prove that this was not the very single time you give, you have the result. So these three compounds should form your layout for experiments. And then you go to the results and you obtain data, and then you lose a lot of time on processing and analysis. So this results part is kind of independent from your layout. But when you're on layout stage, you have to think whether the results you are going to obtain is really possible to process, and whether you have the capabilities to analyze them, and whether it's worth analyzing. Because if your experiment is simple and then you get the results and then you're not really able to analyze them because you need a kind of half a year to analyze them, maybe you have to step back and think about better experiment layout so that the results you get, they're better and you don't lose so much time to analyze and process them. And then when you get your results and they're processed and analyzed, then we go to the discussion. And the discussion is basically an answer to your task question and something that influences your theory. So as a result of your discussion, you have to find out whether you have an answer of yes and no. So this is how it should look like. Here we are going to talk about the controls a little bit, because talking of controls, it's just controls is different skill. We are going to discuss them shortly now, and then we were going to discuss them more precise on your seminars. I don't know how they are going to be held, like because of this coronavirus, but we are going to talk just a little bit. So in most cases, one parameter, like when you go for the controls, one parameter is one control, at least. Sometimes one parameter needs two controls or three controls, because, for instance, when you apply light, then you have at the same time intensity and wavelengths and maybe illumination pattern, but this is already complicated. So then you need two at least controls, one for the wavelengths and the second is for intensity. So when you add substrate for some reaction, then you have concentration and amount and the volume, probably. These are like three parameters for one action. The same is for the labeling solutions, for instance, when you have your live cells and you want to label them with some dye, and you label them for 10 minutes, and you think, okay, my control is labeled cells and unlabeled cells. But if you go deeper, your control is labeled cells, unlabeled cells, and the cells that are incubated with the buffer with your dye, but without the dye itself. So then you need at least three of them. So on this stage, you have to analyze precisely what you are doing and how you can really describe your action or your influence with changing your parameter, because otherwise you have to like run around and cry, why do you not really have your good results? So for the physical parameters, it's easy way of you add something or you do not add something. For chemicals and dyes, we've discussed also. Sometimes you do not have this yes, no possibility, and then you have to check concentration. Like this is for the treatment, for instance. You cannot just tell, okay, I check whether I have antibiotics and I don't have antibiotics, because then you have to check different concentrations of antibiotics. It makes sense when you start your new set of experiments, I mean, completely new, when you jump into a new area, to lose some time and to sit and to think, because usually then you start with some experimental procedures that are made by people 10 years ago. So you have to think and to analyze what are the parameters in these experimental layouts and what are the control parameters. And do you really need to add some controls? Because science have changed a lot through the last 10 years, for instance, and many parameters that 10 years they were okay. Now they need a control as well. One of the examples, like the one I like most of all, is the DMSO. So DMSO is the solvent you usually use to add dyes or chemicals, or sometimes when you want to treat cells with something, and you use a kind of 1 to 5%, and sometimes it's 0.1% of DMSO. And 10 years ago, it was okay to use it without control, because people told, okay, DMSO starts to influence your results starting from 5%. Now, if you go for the research gate, you see that DMSO really influences your cells starting from 0.01%. And this also depends on the cell type. But if you use the protocols that are developed like 10 years ago, nobody would really mention this parameter and that you have to do the DMSO control. So you have to think about it on your own. And even when you adopt experimental layout from publication or from site, like from Thermo Fisher or Upcom or whatever, then you have to think once again what controls you have to add to this experimental layout and whether it's completely covered by the experimental layout that is copied by you from other source. So good experiment has a control for every question that is asked by some colleague that doesn't really know what you were going to do and what was your aim. So it also connected with the questions the editors will ask you when you try to publish your results, because the controls is the favorite questions for each or every publication, and sometimes it may takes more space and time to make these controls for the experiments and experiment itself. But if your controls are good, then your experiment is good, if the question for the experiment was also good. So here we are back to this bacteria, and here we have a list of the parameters and the controls that are possible to do if you work with some abstract object. Because I do not really know how many of you work with cells or with model animals or with bacteria, and it doesn't really matter so much. So it's good if you have this list for yourself and you make markings of what have been checked and what have not been checked. And sometimes it also depends on the device. you're using. For instance, high humidity and low humidity. If you use a new incubator for the cells, then you, in most cases, you have the marker. So if the light on the door is not really blinking, then everything is okay. For the old devices, you don't have this lamp, and then you don't have this marker. So you have to check your humidity and to write it on your laboratory journal that humidity was okay, because otherwise you do not have the improvement of your good humidity. So stress in behavior for model systems is really complicated parameter, and sometimes you have to make an additional experiments to prove that there was no stress, that the cells were in good conditions, that your mice or fish or rat or humans were okay. And sometimes you need extra treatment to prove it, and you have to think it ahead of doing other complicated and valuable things. But after you've made your pilot experiment, so first you prove that your idea works, and then you start thinking about controls. If you cannot really specify all the parameters, and you cannot really tell which one is controlled and which one is statistically independent, and which one is just something that came from Mars, then probably you have to come back and change your experiment layout or to change your aim of an experiment, because sometimes it appears that you just cannot solve the problem because you cannot really control all the parameters. And then it makes sense to solve some other problem instead of wasting time on solving this one that you cannot really solve. Sometimes it makes sense to look and Google whether these what if parameters were already checked by someone. So then you at least have a plan how to check them. In some areas that are new, it makes sense because otherwise you are trying to think ahead of doing something that was already solved. So you have to be on touch. So apart from these controls that show that your parameters are really controlled, you have to make controls that allow you to get data even if something went wrong. For instance, excess of bacteria in just one test tube, or just factory defect of one test tube, or the sedimentation in the bottle with your buffer or your treatment solution, that all this end volume came into the same test tube. Or the test tube was just on the edge when it was colder, warmer, or there were more light or less light. So if you have these repeats, you prove that your experiment layout worked. And even if it didn't work in like some % of cases, then you still have results from your experiment. So if you had 2 tubes, one went wrong, one went okay, then it's not okay. If you have 10 tubes, one went wrong, 9 went okay, then you have 9 tubes with the result of your experiment. Then the question is asked about how do you distinguish how many tubes are okay? And this is the art of performing the experiment. So the complicated experiment need to have more runs. The more runs you have, the more expensive your experiment is. The more expensive your experiment is, the less number of experiments you are able to perform in most cases. So you have to balance between how many tubes you can lose and how many tubes should be okay, and what is the percentage of the bad tubes, because if you have, for instance, 20% of tubes where something went wrong, then you need to go back in your experimental plan once again and check whether your experiment layout is okay, because 20 or 25 or even 10% is not really okay. So a competent experiment gives you a sufficient number of results, which in this case means that sufficient number of tubes are okay and just one is spoiled. So to summarize, our plan was about experiment parameters and then controls and then replications or repeats, and then it's going to be about results and qualitative and quantitative and measurable and not measurable parameters, and then analysis and statistics and probably models and conclusions. So you have to repeat your experiment so many times that you have sufficient number of the results. And this leads you to the statistical parts of result area. And here you also have to Google and to think about how many runs is the statistics. Because sometimes you have the publication and you see that the number of experiments were 2, and then there were kind of 100 technical runs within the experiment, which is sometimes acceptable. But sometimes it's not acceptable, and you need to have hundreds of experiments and just 2 technical rounds within the experiment. So this is going to be the second question for take home. How do you really distinguish how many runs and how many repetitions you have to have? And what is the difference between independent and non-independent experiments? So what is the independent experiment in this case? For instance, for cell culture, you have your aliquot with frozen cells in most cases, and then you take it from the fridge, and then you see them, and then you care for them, and then you make an experiment, and then you take the rest of the cells and take care for them, and they're growing, and then you repeat your experiment. Whether this is an independent experiment, should you count it as independent? For instance, you have your mice, and then you have your mouse pups from 1 mouse pairs. Whether these 6 mice are independent experiments, or they should be counted for repetitions, or whether it's gonna be counted as technical replicate. So I want to hear from you, like next week probably, and it would be great if it is going to be in written form. So what is the technical replicate? What is independent experiment? And what is the run? What is the difference between these 3 things? Okay, so we are going to talk about limits you have in your experiment, and there are kind of few different types of them. So they are logical. Logical is mostly what we discussed previously. It's the layout and repeats and parameters that you can control, and some of those that you really cannot control, like weather outside, for instance, and the values that you are gaining during this experiment, and probably device limitations. So if you're not really able to see the wavelengths 650, then you probably don't need to make the experiment. And the time restrictions. So the time restrictions in this matter is whether your results are good, whether it is possible to analyze them, and whether it makes sense to analyze them. All of those are logical restrictions. So for ethical restriction, you have the replace, reduce, refine, so 3R procedure that you should remember. This goes when you work with animals and model systems that are more complicated than cells. And sometimes it goes with your, also with your experimental layout if you work with humans or close to humans. You have also to remember about ecological 3Rs, which is reduce, reuse, recycle. And these ecological ones, they go closer to the financial. So when you have to use minimum to get maximum, which goes back to the idea of controlling and replicating, and the question about how many tubes you have to have and what is too many. So you have also to remember that you need to produce valuable, needed, and actual results, and there should be demand on this knowledge. In Russian, it's called that the science is a way to get answers to your questions and be financed by the government. It's not like that in all the countries, but you have to remember that you got financed from the people that are not really interested in you knowing answers. They are interested in them improving something. Therefore, your results should be on demand. They should be needed and they should be expected. Because also sometimes when you have your results, but they are not expected, then there is still no value in those, although they do really answer to some theoretical question. So the last but not the least, what you have to remember is that your experiments, they should be conducted in such a way that anyone could justify them and repeat them. So this goes about describing your experiment, and this goes also about laboratory journals. I'm not really sure that you still do it on written form, not on electronic device, but still. So all the logical, ethical, and other limits of your experiments should be described in your lab journal. All the procedures and all the parameters you thought about should be described in your lab journal. Sometimes these are parameters that you do not really think about it. For instance, when you work with antibodies, you have to write the batch, because sometimes when your like tube is empty and you go to the new tube from the same manufacturer, the same type of antibodies, everything the same, but different batch number, you have different results. And if you do not have it written on your lab journal, then you never see why was it. The same goes with cultural media, and the same goes with mice photo. And the same definitely goes with some treatments. And it's a little bit funny or a little bit sad, but for some chemicals, you have to write down how many times they were thawed and frozen. So the cycle number, because it makes difference. So it's better if you just write down everything and just try to pretend to be paranoid and write even things that you do not really think are important, at least when you are in the very beginning of your experimental work, because sometimes it makes a difference whether the elevator was working or not. So these examples that I'm just going through, they are real examples from the real labs and real experiments that influenced the results. It's not that I'm just making my fantasies. So your lab journal is improvement for your experimental layout and improvement for your results. If your lab journal is bad, this makes your experiment not valuable, worthless, and not really useful. So please remember about it. We were going to discuss the lab journals on our practical part, but I don't really think we are going to have that, not this month at least. But we can discuss it if you have questions. Usually you do not. But if you want to discuss it, we can go through it. So I was going to talk about a little bit about analysis, statistics, models, and type of results. I'm not really sure whether you're interested in it, but please, so about the statistics, please, before you conduct your experiment and before you start to perform your physical actions, go through the articles and look. What type of statistics do you need for your analysis, and what type of statistics was already discussed? Because sometimes the experiment is okay, and the number of controls is okay, and number of replicates is okay as well. But still, it's not enough for the statistical type you want. Sometimes you need big data to prove your theory. Sometimes for some statistical criteria, you need like something like more than 1,000, and you have 900. Sometimes you need, for instance, continuous signal, but not signal in different time points. And then you're not really able to apply statistics that you were going to apply. So please think about it before you start your experimental work, because it's really unhappy case when you have done already everything and still you're not really able to analyze it the way you were going to, because you didn't really check what are the demands on the statistical algorithm you are going to apply. The same goes with models. So if you want to use a model, especially if it is mathematical model, please check before you start what are the parameters of the models, whether you're probably able to use this model with your experiment. Because sometimes it's really sad when everything from the laboratory and from this biological point of view, everything is made correct, but still you're not really able to analyze it. Please think about the amount of work that is going to be done to analyze your data, because sometimes the experiment is okay, but you have not got the automatization and you don't know how to analyze it later on, because you have, for instance, 10 terabytes of data and it's in graphics form and you don't have time. So this part also goes as experimental layout. Please plan how many days, nights, weeks do you need to perform the analysis, because otherwise you're not really able to publish it on time and then you don't have a profit. So here we are not going to go through discussion, because it depends on the journal you're going to publish, and depends whether you're going to do it as a diploma or are you going to make it like something else out of it. So the forms of the discussion are different, but we went through the plan of the experiment briefly. we were going to have a practical part on this, and I still think that it would be good to have it. So I ask all of you, I think we're 9 right now. I ask you to write your imaginary experiment in your area, like the area you are going to, or you want to study in. Just imagine that you have all type of the instruments, all type of the reagents, just everything. But you have only 4 months of time. So everything you can imagine, and 4 months of time, and I want you to make a plan of the experiment, imaginary one, and to predict the results, and to write a plan on how much time would it take you to do this part and that part, and how you're going to analyze it, and what are the controls that you really need, so that we could go through and just discuss whether it's possible to do such a thing or not. So this is mostly it. So what I want also to ask you is, do you want to have a practical part in any form so that we can think about it? Or do you want to have these lectures? And is it okay for you to have the lectures like that? Or do you want them, for instance, to be recorded and then published somewhere so that you can have the access anytime you want? Or what is your wish list? And that was the question. So I would be grateful if I have some answers. No demonstration.
All right, bro.
The classes
Hello,
should be
bro.
recorded. Okay. Any other ideas on what I've asked before?
Yes, I also support the idea of the class being recorded and posted somewhere that we can have access to it afterwards.
Okay,
At
what
least
about...
it will help us. For some of us that are having network or internet challenges, it will help us to catch up after the class.
Yeah, I
and
think
then for the practical...
Yeah. Please go
for
on.
the practical aspects, I also will appreciate if we can do the practicals together as a guide. Then it's a welcome idea now. Thank you.
So for the practical aspects, we can discuss some of the ideas. We cannot meet like in reality and do the practical aspects, but we can discuss something. For instance, we have in some lectures, we are going to discuss practical use of some methods, for instance, advanced microscopy, and this can be discussed in practice, like solving practical and discussing practical things and discussing real protocols. Or what we can also do, and I find it more practical, is that we do a journal club in such a way that after each lecture that is dedicated to one method or some area, we take one article which is really important in this area and we discuss it. Like, imagine that this was our practical work, and then we are going to discuss what was good, what was bad, why statistical measurements were made in such a form, why these parameters that are published are important. So, because this is less imaginary, because it's based on real work. And still it's a little bit of practical, because then we discuss something that was made by real people and makes sense.
I think that is a good idea.
So if it is okay
Makes
for
sense.
you, yeah, I think that makes sense. So then please, I need
Yes,
at least until because next week I'm not sure that we will manage, but until next week I ask you, those people that are interested in this journal club, I need your emails so that I should send you an article, and you have all of us have 1 week to read it and to think about it. And then the week after next week, we are going to discuss it as a practical part of our course. But then I need to know how many people are participating, because I'm going to share this article and figures from this articles. So for instance, one person is responsible for describing one figure. The second person is responsible for describing second figure. It doesn't really mean that you do not read all the article. It just means that you have to be prepared to discuss this particular part, and probably you Google through the questions you already have, so that you could help other people that are not really sure about what is on this picture and why this picture is important and why was it included in the article. So therefore, I need to know how many of you are participating. It's not going to, there are not going to be any restrictions on people that don't want to. So please feel yourself only in case if you are interested. Yes, my email, I think my email looks like that. Just a moment.
Hello, Ma. I have a suggestion.
Yes.
I think maybe a WhatsApp page or a Telegram page can be created to unite all of us that will be participating. So any information you want to pass across to us can be actually channeled into the page, so for all of us participating to see.
That would be great, but then I need your names in, so then I need you to be self-organized in some way, or just tell me whether you want to have Telegram or WhatsApp, and then I need your names or how do yes, because for me it makes no difference whether to use WhatsApp or Telegram right now. We can use both maybe, but then it would be a little bit slow, because I can forget that I have 2 of those, but you have to check one and to choose one that we are going to use.
Okay.
So you can, if you want, you can write me an email and just tell me, okay, I want to participate and these are my names in WhatsApp and Telegram, and I prefer this or that, so that I summarize it and okay, then choose one. What we can also do, I think it makes sense, I will do it. We are going to have a folder in Google Documents so that the articles are there, and maybe the protocols that we are going to discuss are also there, so that you could place them there. Please, this is the thing I forgot to mention. So when discussing our protocols, please remember that if you have some secret data, do not mention it there, because what we are doing is public. I don't think that this is the case, but sometimes people, they are a little bit tough with that. So what we are doing here in class, it's all public. I'm not going to publish it for you, but it's not going to be like private or something. So if you have some specific things that nobody have to know about. Please remember that I'm also nobody, and probably your colleagues here are also a bit of nobody. So just remember. Okay, any other questions? But still, since you have your email, you can ask the questions by email as well. You're most welcome. Sometimes I'm a little bit slow with answering, but I will answer the emails. Anyway, please remember that there were 3 questions during this lecture, and there was 1 mistake on this presentation made on purpose. So we were like close to this mistake, but we didn't really discuss it. So it would be good if you just go through the presentation 1 more time and think what could be the mistake there. It's not in the type of experiments. No, it's not in the theory. It's a practical mistake, and I would be glad if you find it. Okay. So then basically we see us next week, the same time. Yes, I will write the home questions in the chat form just in few moments, but then I have to stop talking because I'm not really good at talking and writing at the same time. So then 5 minutes for me to write the questions and for you to think if you want to have something else. So for the last two classes, that didn't go as we planned. What we are going to do is, since we decided that it's okay for you to have the lections written and shared, so I think then we write them down and publish somewhere. Most probably it's going to be YouTube or something like that, and you'll have the access. So I'm not really sure when it's going to be done, probably until the mid of November we are going to do this, because one lecturer is not really in Moscow right now, so we have to wait until for him to come back, her to come back and then write this lecture. But still, I think in 2 weeks we are going to be on schedule with our lectures published. Okay, now I think we are done. So as soon as you send me your emails, I will send you something back. But now I think we will close this all. So when I close the conference, you'll lose the message board. So if you want it, please copy it right now. You have kind of 5 to 10 minutes to copy it, and then I'm closing the conference.